important invention for us, at least, working with ancient DNA is this emulsion PCR, because this allows a single DNA molecule to be amplified without being in competition for the polymerase, for the nucleotides with the other DNA molecules. And so if you have this mixture of damaged old DNA and undamaged modern DNA, then the modern DNA always amplifies much, much faster. And then this is the reason why it is so hard to sequence up the ancient DNA. And one trick that we used is traditionally the ancient DNA work is all done by drilling holes into bones, and from the bone powder you extract the DNA that is bound to the bone material in making phosphate links. And the disadvantage from those bones is that they are very porous and they have an almost infinite surface. And in this process of an animal degrading the putrefaction, the animals that live, the bacteria that live in the gut of the animal, that as the biomass is going away, they dig themselves deeper and deeper into the bone, and eventually they die and deposit their own DNA on top of the DNA, for example, from the mammoth. And this is not happening if we use hair, and it wasn't even clear to us that there would be so much hair from extinct animals. This one here I bought on eBay, and this was before people even knew that this would be meaningful. It was $130, and the prices for mammoth hair since our study has gone way up, so I should have bought a lot more before I published this. Particularly if you consider a mammoth would have a lot of hair, and this is something that we need to come back to in a moment. But in the moment we realized that hair is a good source, we contacted museums. And by the way, I do not encourage anybody to buy archaeological finds or specimens like this off eBay. The way that we actually did it after we found out that this was a vendor, that we asked them to provide us with a valid export permit, and we also made sure for the university system that this was a valid vendor. So you should not go crazy and buy and create a black market for exporting those things. And this is why we truly worked with all the museums in the world that we could find. Some of them were sending the last hair that they had. And what this allowed us, so I tell you the trick that why we are so crazy about hair, and that is that the hair, this is a cross-section through a mammoth hair. You have the medulla, the cortex, and the cuticle on the outside, and the keratin is like a biological plastic. And unlike the bone, it's not porous, and so the keratinocytes that are forming that hair shaft, they die in an apoptotic process. And then the nuclear genome is being chopped up into small pieces, but it still is encased here in this biological plastic. So we take the hair, we shampoo it, and then we bleach it, not to make the mammoth blonde, but to decontaminate and destroy the modern DNA that is on the outside of the hair shaft. So that we destroy with bleach, then we neutralize, and then use proteinase K, and we dissolve the biological plastic, and the mammoth DNA on the inside is being released. And so when it comes to the question whether bone is a better source or even this dried up meat, just the last few days, again, there was this announcement from the group from Japan. They always do this every 12 months or so, where they say, we have found this amazing bone with bone marrow, and we're going to clone a mammoth using an Indian elephant. But if you've seen through our work that this entire set of chromosomes is being completely shredded into pieces, there is nothing in there that is longer than 100 or maximally 200 base pairs. And so this claim that these people make is completely unsustainable. So don't hold your breath to see a mammoth cloned in the next few years. And so this discovery that this keratin approach is so good also works for the horn, and it also works of hooves, of beaks, of claws, whatever is keratin is great. And now what can you do with it? Before I go there and I close this section, I want to tell you about our museomics. Museomics is museum genomics. And why did we come up with this idea? This is the Adams mammoth, the most complete found mammoth skeleton to this date. It was discovered in 1799, and believe it or not, it was with this find that people understood that animals go extinct. Because until then, it was pretty much decided by religious thinking that it was up to God whether a creature existed or not. But this was the first time that people would say, why are the elephants at the polar circle? And these elephants are not like the Indian elephants, they're not like the African elephants, so there must have been a species of animal existed at some time, and it's not existing anymore. This was so exciting that the botanist, Michael Adams, who found it, hit the Tsar up for money. It's like today if you do an NIH application or so. And he got the money, and 3 years on horseback and sledge, he went to Siberia. They dug out that, brought it back 3 years. It arrived as a big stinking mess, and the Tsar was delighted. Apparently his wife wasn't so much. She didn't like the smell. But what was amazing is what a transforming discovery that was by unearthing, unraveling those mammoths. Together with that skeleton, they found 36 pounds of hair. And they graciously sent us 0.1 gram of that hair. And from this 0.1 gram, we were able to sequence a complete mitochondria. And this is now shown here. This is one what we call internally in the lab. Feng Xing was all with us at the time when we were doing this. We call it the chi figure from his colleague, where we do a graphics that we show you all the genes. So this is the mitochondrial genome from base pair number 1 until 16,800. These are the genes as they are annotated. And for every difference to a reference, if you compare mammoth 1 with mammoth 2, 3, 4, and wherever there is a sequence difference as a single nucleotide polymorphism, we make one of those bars. And I want to train you through the rest of the talk that you become experts yourself in looking at the biodiversity of species. What you'll see here is that everybody is pretty much the same, right? Except for 3 guys. And those 3 are very similar to one another. And this was this figure that gave us the idea that there must have been a second group of mammoths 45,000 years ago. And this was then calculated, and I'll show you the Bayesian analysis in the next slide. But before I go there, this is this orange bar that I want you to look at. This is the hypervariable region that includes part of the SIDB gene and the non-coding D loop. And traditionally, only the few SNPs that would be in here would be analyzed. The other important thing is, so this group, what we call clade 1 of mammoths, they have roughly 20 SNP differences between 2 pairwise mammoths. But I need to remind you that this is the diversity not of all the animals living at the same time, like today, like the diversity of us. It is a third dimension because those diversity is spread out over 50,000 years. So if you take this into consideration, this is a very, very low genetic diversity. At the same time, where we stand and why I still think that this mitochondrial genomics is very important is, if you look in the database, even today in 2011, there is a single complete mitochondria from an African elephant. There is a single one from 2 from an Indian elephant, and through our work we have 18 from mammoths. So the extinct elephant has more than the living ones. If you take your favorite deer, whatever population you have out there, there is no mitochondria of a deer in the database. So there still is plenty to do, and this is the message here. If you are looking for a project, go sequence mitochondria. If you take those mitochondria and you do a Bayesian analysis, and then the 3 individuals that are so different are here clade 2. These are all the individuals that make up clade 1, compared to Asian elephant, compared to African elephant, compared to mastodon, another extinct elephant. And by that we can then very precisely calculate the population split times, and this is why I'm relatively confident that these 2 mammoths separated from one another about 1 to 1.5 million years, and they died out